So to start off our meeting, we're very happy to have Dr. Adam Moeser with us, and he comes from us from Michigan State University. He flew in late last night, and one of the things that we want to talk about was gut health, and that's what Dr. Moeser spent his time researching and working with in different capacities. But right now he's with Michigan State University at the College of Veterinary Medicine. So I'm going to get Adam to talk a little bit about more of what he does and his background a little bit. But we welcome you to Toronto, and we're really glad that you're here, and you get to kick off our meeting. So good morning, and the floor is yours.
Okay. Well, thank you very much, and appreciate the opportunity to be here. Talk to you a little bit about what we do in the research lab, and overall, this is going to focus on gut health, and a little bit about what we'll talk about here. But first of all, maybe give you a little background on myself. So I'm originally from Massachusetts, and my graduate work in North Carolina. I did my master's degree in pig nutrition at North Carolina State University, then went on to do a combined DVM PhD program. And in my PhD program, I focused on gut physiology, and I became really fascinated with the GI tract and how this was so important, the function to health of not only animals but also humans. And then started on faculty at NC State in 2008, and then since last year we made the move to Michigan State University. And the major goal or objective of my research program is to investigate how stressors in the environment of animals impact gut function. And we take a very basic physiology approach to understand gut under various conditions, and it's our hope that as we learn more about the basic physiology of the gut, we're going to be able to develop more therapeutic strategies to target this important aspect of pig production, which is the effects of stress on GI health. For this talk, I'm going to be focusing on a couple different areas. First, I'm going to be covering some very important aspects of gut function and health. What's the machinery that protects animals in a daily productive environment from getting disease? And also talk about some of the differences between the versus nutrient absorptive function. Then I'm going to talk about how these functions of the gut become altered and under stress conditions here, but also how they can be changed in diseases of enteric pathogenesis. And I'll cover a bunch of different diseases, but then as I go on in the talk, I'm going to be focusing mostly on enterotoxigenic E. coli and some of the work that we've done with post-weaning E. coli. What are some of our new understandings and the insight of the physiology that happens when an animal is exposed to this particular pathogen, and what are some of the short-term effects as well as the long-term effects on gut health. So to start with the gut, here's a picture, a little histology figure of the GI tract of a pig. It could be any animal, and you can just see this. It's lined by this single layer of epithelial cells. And this epithelium and underlying mucosa, the function of this is critically important. It serves divergent roles. On one hand, it must absorb nutrients and transport water and electrolytes, but at the same time, it must serve as this selective barrier against the outside world, because the gut mucosa is the largest interface between us and the outside environment with the surface area of the villi. And this becomes critically important if you think about it, because you have this single layer of epithelial cells, and millions or trillions of toxins and antigens normally that reside in the intestinal lumen, which is literally micrometers away from the largest immune organ in the body. So how this gut functions to preserve this barrier while at the same time absorb nutrients, it's a critically important function, and that's an area in my lab that we study. We study how this barrier function is regulated and how we can protect it during stress. So if you take a closer look at the intestinal barrier here, this is the gut epithelial cells lining the GI tract. There's many factors in the gut that protect animals and us from disease. There are some extrinsic properties such as protective secretions from the gut. Chloride and bicarbonate are very important. There's specialized cells such as mucus cells that secrete mucus that forms a protective layer which has many functions. There's specialized cells such as Paneth cells that can secrete antimicrobial peptides. We also are learning a lot about this microbiota, this resident bacteria, which confers very beneficial properties as well. And without this or disruption of this microbiome can lead to increased risks of GI disease. There are also some intrinsic components of this barrier, and these include the tight junctions. And tight junctions are proteins that are located in between the intestinal epithelial cells. And these are critically important because they regulate how leaky the gut is. And they often become disrupted or broken down during a lot of enteric diseases or stressors. And once these break down, you get increased intestinal permeability. Another important component is this immune system here that is in the gut. And this includes factors such as secretory immunoglobulins as well as resident immune cells that must be ready to, if there's any breach in this intestinal barrier or leakiness, these immune cells must be ready to respond rapidly to prevent the spread of infection. So optimal functioning of the immune system is part of this barrier. And then I also have the nervous system and the circulation, because a lot of these functions, barrier function, absorption, secretion, are regulated by the nervous system. And what we've been finding out in the laboratory in some of our research studies is that during a stress, the nervous system is always activated, the intestinal nervous system, and this is driving the adverse changes that occur in a stressed gut. So what happens when this is broken down? So when it becomes disrupted, you get loss of tight junction proteins, and you get increased intestinal permeability. This allows unimpeded access of antigens and pathogens now to get across this gut. So they're in this leaky gut situation. Also, what can happen is this: once these pathogens get triggered, they start causing inflammation in the gut, and this in turn can impair nutrient absorption. So not only do you have a leaky gut and inflammation, now you're getting a compromised ability of the animal to begin to efficiently absorb nutrients. And if this, and this also can contribute to some secretory aspects. And there's a lot of processes that occur, a lot of immune cells that are activated that drive fluid across the gut, which to diarrheal disease or loose stools. Now, if this barrier becomes chronically leaky and there's persistent activation of the immune system, what can happen is that inflammation is then going to give over to immunosuppression, because the immune system can become overwhelmed, have chronic antigen stimulation. So conditions where you have chronic leaky gut, and I'll go into how certain stressors can affect that, then the immune system is not working as efficiently anymore. And then you have increased susceptibility to disease. And another part of this that we don't talk as much about is the ability of this gut function to repair, because there are certain times where the gut is going to be injured. There are different stressors, there's different challenges, but an important part is recovery and understanding. So the gut must be able to rapidly recover and restore normal barrier and absorptive function after challenges as well. So how quick the gut can do that is very important as well. So now I'm going to talk about some of this gut functional changes. I'm going to talk about how these specific aspects of gut function can change during an enteric infection. So really going to cover sort of enteric pathogenesis. So talking about E. coli, we know that E. coli is a secretory, it's classified as a secretory diarrheal disease. And this is a disease where the functional defect in the gut is that there's just hypersecretion. There's too much secretion leading to diarrhea. And here's a cartoon or a picture here of the intestinal villus of what happens during E. coli infections. We know that E. coli can elaborate a number of different enterotoxins that can bind to the intestinal epithelium and trigger the cells to oversecrete. A lot of chloride, a lot of bicarbonate, and then water follows, and this drives the secretory diarrhea process. So a normal process in the gut, important, now becomes sort of hijacked by these enterotoxins to increase and drive secretion. One thing that we always talk about E. coli as a secretory diarrheal disease, but the toxins can also influence these absorptive epithelial cells as well, because some of these same toxins and the signaling pathways that they initiate can also inhibit sodium absorptive processes of the gut. So during an E. coli infection, you're getting hypersecretion, but you're also getting impaired absorption. And towards the end of this talk, I'm going to go a little bit more into some of the work that we did with absorptive capacity in the gut of an E. coli infected animal. Another type of diarrheal disease is associated with impaired absorption, so absorptive diarrheal disease. And this is classically with diseases that are like viral associated diseases where there's some marked changes in the morphology of the intestinal mucosa, where you get villus atrophy from the infection, you get loss of epithelial cells. This decreases the mucosal surface area, which in turn is thought to impair the nutrient and water absorption, leading to a malabsorptive diarrhea. And these are, like I said, pathogens such as PED, TGE, rotavirus are commonly associated with this, as well as other diseases that can affect the mucosa, such as coccidiosis and dysentery as well. But it's also known that some of these viral pathogens can also cause hypersecretion in the gut as well. They secrete some viral toxins that drive secretion as well. And then there's other diseases which we talked about the immune response. Well, this is Salmonella, for example, is an inflammatory diarrheal disease. And this is a situation where there's a hyperactive immune response in the gut. And this is what's responsible causing disease. So salmonella is a pathogen that binds to the surface epithelial cells, and it'll generate a number of different cytokines and chemokines being released by this epithelium. This recruits a ton of neutrophils into this tissue, which are the inflammatory cells, which cause the intestinal damage seen with salmonella. But at chronic conditions, you can start seeing some major changes in the morphology of the gut, as well as some other immune cells, adaptive immune cells, coming into the tissue under a chronic condition. So now, under these conditions, you're getting malabsorption, hypersecretion, and also some permeability defects as well. And this is, I'm sure you've all seen this. This is just a slide of what salmonella looks like. This is just a healthy control ileum. And then this is what it looks like under a salmonella infection. You can appreciate all the increased number of cellularity, neutrophils, and basically the tissue looks like a large intestine. It's the villus has been atrophied, and just a tremendous reduction in surface area and absorption. So just to recap and summarize the first part of this talk, we know that the GI tract has many critical functions. It must perform digestive functions, barrier function, immune function, must eliminate waste, and must also be able to rapidly repair and regenerate after an infection. These are all very important parts of the gut health. And we know that when the gut is under a challenge, such as a pathogenic challenge, there are some of these basic gut physiologic processes can be dramatically altered and by these pathogens, and this is what's leading to disease. So now we kind of have an understanding of some of the basic parts of gut function and how they can be influenced. I'm going to go into some of the work that we've been looking at in our lab, which is the impact of stress, stressors in the environment, and how that impacts the gut health of an animal. So we know that many diseases, including especially gut diseases, they're really a result of interactions between the host, the pathogen, and the environment. And we know this because we have the host, which are often in pathogenic environments, including us. We live in a pathogenic environment. But we know this relationship alone is insufficient to cause disease because animals are in pathogenic environments and they're not sick. So are we. So, and this is because of these defense barriers that are held up and prevent us from infections in a normal environment. But it's not, and there have been studies shown that there's one of the major factors that are going to determine whether an animal gets sick or not is these environmental factors. And you all know this, is that the environment in which these animals are in can have a tremendous impact on whether or not this animal or person is going to get sick. And although there's been a lot of research focusing on the pathogenesis here, very little is understood on how these environments can impact the host to cause increased susceptibility to pathogens. And this is a particular area of focus for my research laboratory. And one of the major factors that we know is a susceptibility factor in disease is stress. All these stressors in the environment are often preceding the onset or breaks of different diseases. And we are really interested in understanding the physiology behind that. So we've known that stress is a trigger of disease in animals and humans. We've known this for a long time, and stress can manifest itself as poor weight gain, susceptibility to GI infections, as well as more complex GI disorders such as inflammatory bowel disease, as well as irritable bowel disease. These are more of diseases that are associated in humans, but we're also finding that some of the stressors that animals undergo, they can look remarkably similar to human diseases as well. So the effects of stress on the GI tract in us and in humans, and in animals, are quite, and we're seeing some of the similar findings. But we've known that stress has such an impact on gut function. We've known this for a long time. However, really the mechanisms or our understanding of the biology of how stressors cause disease still remains poorly understood, despite this importance of it. And we know that there are a number of stressors that animals undergo under a normal production system. And all of these have potential to have an impact on increasing disease susceptibility. So this is as I had mentioned, that this is really the focus of what we study. We're very interested in how stress impacts our guts and how this increases GI disease susceptibility. And we're interested in a couple different areas. We're interested in early life stressors and GI development. I'm going to talk about a weaning stress and how the age of weaning can impact long-term gut development. We're also interested in some of the underlying mechanisms and how stress can impair the barrier or immune function of the gut. And also, what can we do with management, nutrition, and the interactions of these to mitigate stress-related diseases.